fixation. So they are used as biofertilizer. Okay, biofertilizer. They increase soil fertility. They increase soil fertility. Now in India, okay, in rice field, the cyanobacteria which is used in maximum, the name of that cyanobacteria is Olocida. Okay, Olocida is maximum used. Okay. And for maize and sugarcane, the cyanobacteria which is used, its name is Cylindrospermum. Okay, Cylindrospermum. Now, some cyanobacteria are also used as single cell protein. Okay, single cell protein means they are used as a food. Get protein from that. Example is spirulina. Spirulina is a blue-green algae. Okay, and we can say that they are used for nitrogen fixation as well as carbon dioxide fixation. Okay, so with this we have completed cyanobacteria, students. With this we have completed cyanobacteria. Okay, our next today we will start Mycoplasma, students. Okay, Mycoplasma. Now, Mycoplasma was discovered by Nocard and Revi. Okay, it was isolated from cattle suffering from pneumonia. Okay, pneumonia due to absence of cell wall. They can change their shape. So this type of bacteria is called, so it is called pleomorphic bacteria. They are also called PPLO. Okay, full form of PPLO you should know. Pneumonia like organism. They are also called jokers of plant kingdom. Okay. Now Borel gives the term, students. Borel give the term mycobacteria Asterococcus. Okay. Asterococcus. Okay. Novak give the term Mycoplasma. They are also called MLO, that is molecule like organism. Molecule, why they are called molecule? Because they have soft body. Why they have soft body? Because cell wall is absent. Cell wall is the rigid structure and that is absent. They are also called coat of organism. Coat of organism. Why? Because cell wall is absent. Now they are resistant to penicillin. Why they are resistant to penicillin? You know the reason, friends. I have discussed with this already. Penicillin only act on the cell wall and they don't possess cell wall. They are resistant to penicillin and bacitracin because cell wall is absent. But they are sensitive to streptomycin. They are sensitive to streptomycin. Now here cell membrane is present. Here cell membrane is present. They have stabilized sterols. Now this feature is sterol is a feature that resemble eukaryotes. In eukaryote, sterol is present. Okay. Like in bacteria, mesosome is present, but here mesosome is absent. So who will help in replication? Suppose this is double-stranded DNA. Okay. Now this double-stranded DNA is linear. Again, it is a characteristic feature of a eukaryote. Eukaryote characteristics, because in bacteria you have seen circular DNA is present. Okay. So this, suppose this is the chromatin part or this is the chromosome part. Now in this chromosome part, one disc-like structure is present. Okay, one one disc-like structure is present. Okay, it is called replicating disc. Okay. This structure is replicating disc. Okay. Now this replicating disc actually help in replication. It help in replication because mesosome is absent here. So we can write here that mesosome absent. So its function is performed by replicating disc. 70S ribosome is present. 70S ribosome is present. All prokaryotes, student, they contain 70S ribosome. Eukaryote 80S ribosome. Now its one feature also resemble with virus, students. Okay, it's one feature. Like two features they resemble with eukaryote. One feature resemble with virus. What is that feature? They can pass through bacterial proof filter. Bacterial proof filter which will make through that student viruses passes and mycoplasma can also pass. Now smallest mycoplasma is Mycoplasma gallisepticum. Gallisepticum. Now this is the smallest mycoplasma. Smallest mycoplasma. Now if we see the respiration part, student, they are facultative anaerobes. Okay. Facultative anaerobes. What is the meaning of facultative anaerobes, student? Suppose this is a test tube. In this test tube, in the starting part, oxygen concentration is more. So you will get the bacteria here also, but in the interior part, oxygen concentration is least. Almost no oxygen is present, we can say. So these bacteria can survive in presence of oxygen as well as in absence of oxygen. This type of bacteria is called facultative anaerobe. So suppose grow it on a neutral medium, okay, and just allow it on a petri dish. What you will see here? You will see here a colony. Here an opaque colony you will get. What type of colony you will get? Opaque colony. And here you will get translucent colony. Okay. Now this will give a structure like fried egg colonies. This type of colony is called fried egg colony. See, just it is looking like pouch of an egg now. In the center yellow part and that translucent part. Okay. Till this clear, students? Till this any doubt? Any one of you? Students? Reproduction part.
No, ma'am.
Asexual reproduction. Binary fission. Okay. Now they are pathogenic both plant as well as animal. Okay. So we have completed with Mycoplasma students. Our next topic is Actinomycitis. 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 They possess the feature student both with the bacteria as well as fungi. Okay, bacterial features. They are prokaryote. Okay, they possess nucleoid. They are gram-positive bacteria. Their cell wall contains peptidoglycan. The fatty acid which is present here, the name of the fatty acid is mycolic acid. Okay, now this mycolic acid is pathogenic in its structure. They are pathogenic. Why? Because they produces toxin. The name of the toxin is mycosoids. The name of the toxin is mycosoid. And fungi feature students, they produces spore. What kind of spore they are producing? One is conidia. Another is sporangiospore. And third one is oidea. These three types of spores are produced by it. Okay. So this type of actinomycetes are called mycelial bacteria. Mycelial bacteria. Okay. Or ray fungi. Why the word mycelium has been used, student? Suppose this is a hyphae of a fungi. Okay, this is hyphae of a fungi. This hyphae aggregation of hyphae produces a structure called mycelium. So they produce mycelium. That's why this actinomycetes is also called mycelial fungi. What is the importance of this actinomycetes, student? Again, they play an important role in nitrogen fixation. Okay. Importance. They play important role in nitrogen fixation. One example I can put here is student about Frankia. Okay. Now this Frankia shows symbiotic relationship with root nodules of non-leguminous plant. Non-leguminous plant. Okay. Now with some angiospermic plant, they are also showing symbiotic relationship. What are those plants, student? Example we can write here. One is Mirica. Second one is Alurus. Third one is Casuarina. And with some gymnospermic plant also, they show the association. Gymnospermic plant. Example is Ginkgo. With this eukaryotic plant, with this plant, they show nitrogen fixation. Okay. Now greater than 100, more than 100 antibiotics are made from fungi, student. Okay. Both narrow spectrum as well as broad spectrum antibiotics. Greater than 100 antibiotics are prepared through it. Okay. The first antibody which was prepared, that is penicillin. All of you familiar with this word penicillin. Okay. With the fungi called Penicillium notatum. Okay. Penicillium notatum. Notatum. This term was given by Fleming. Alexander Fleming. Okay. Alexander Fleming. And second one. First, this is the penicillin is a narrow spectrum antibiotic because they act only on gram-positive bacteria, narrow spectrum. The first broad spectrum antibiotic is streptomycin. Now, so first broad spectrum streptomycin. This is the first broad spectrum antibiotic. It was prepared with a bacteria called Streptomyces, and it was discovered by Waxman. Okay. Now, till this clear to all of you students? Till this, any doubts? Any one of you? Any one of you? Students? Any one of you? Our next topic is Archaebacteria. Now, they are primitive bacteria, students. They are gram-negative. They are the living fossil because they found in the primitive environment. They are primitive organism without any changes. They are primitive organism without any changes, and they are found in extreme condition. Which type of extreme condition? All conditions we will see. Okay. So let us discuss some comparative study of Archaebacteria and Eubacteria. Archaebacteria and Eubacteria. Now, in Archaebacteria, cell wall is made up of pseudomurine. Okay, pseudomurine. And here, cell wall is made up of murine. Other name of murine is peptidoglycan. Okay. Now, this peptidoglycan is made up of NAG and NAM that I have already told you in my previous lecture. NAG and NAM. Now, the sugar which are made here, that sugar contain L sugar or D sugar. But in Archaebacteria, okay, the amino acid is actually not sugar, students. Wait, it is amino acid. Okay, it is not sugar. It is amino acid. The amino acids which are made up of, they are L amino acid and D amino acid. They contain NAG and NAN. Okay. Now this pseudomurine, but this pseudomurine consists of only L amino acid student. No D amino acid is present in Antibacteria and NAM is absent. Okay. Instead of NAM, they possess N-acetyl talosa muramic acid. Okay. N-acetyl talosa muramic acid. Okay. Now the next thing is that this is the first point with respect to cell wall. So let us see some more points of differentiation. If we see the second point, students, again regarding cell wall, their plasma membrane. Suppose this is a fatty acid. Okay. And again, this is a fatty acid. Okay. So this is a branched fatty acid. We can see from the diagram that it is a branched fatty acid. Okay. Now this linkage, they are made up of ether linkage. Okay, they are made up of ether linkage and their carbon are long chain carbon, like 20 carbon chain, students. This 20 carbon chain are called phytanyl. Whereas new bacteria, this is the head region and this is the tail region. This is head of fatty acid and this is tail of fatty acid. Okay. Now here the linkage is ester linkage. Again, one point of difference, ester linkage. And here fatty acids are unbranched. They are not branched, students. And they make bilayer. How bilayer of fatty acid look like? See the diagram. I've shown it in my earlier lecture, but again I'm showing it to you. So this is monolayer, student, one layer, and this is the another layer. So it consists of bilayer of fatty acid. Now stability wise, this structure is more stable, student. Okay, their membrane is more stable. That's why they can survive in high temperature around 100 degrees centigrade. They can survive on snow due to their membrane structure. They have the more stability, students. Okay. Now, if I'll go to the types of Archaebacteria. Now, what are the types of Archaebacteria? Okay. Types of Archaebacteria. They are categorized into three categories, student. Okay. First one is Methanogens. Okay. Second one, halophiles. Okay. And third one, thermoacidophiles. Now, methanogens means, student, methanogens means they have the ability to produce methane. So what is their habitat? They are found on marshy and swampy areas. They are found on the rumen of cattle. Okay, rumen is one of the component of their stomach of cattle. Actually, these methanogenic bacteria are responsible for cellulose digestion in cattle. Cellulose digestion. Okay. Now, where these halophiles are found? They are found on tidal pools, salt lakes. Now, how much salt concentration? Halophiles. Halophiles means they can bear, they can resist the salt concentration. But what is that salt concentration, student? That is 25 to 30% NaCl. They can resist. Now, some example of halophiles are Halobacterium. Okay. Halobacterium. Okay. Halococcus. Another example. Now, thermoacidophiles means they can resist the temperature as high as around 80 degrees centigrade. They can acidophiles. Their thermophiles means for temperature. Acidophiles means they can resist They can survive till low pH as much as low as 2 pH. And they are also found on hot water springs. Okay. First one is nutrition, students. Second point is nutrition. First one is habitat. What is their mode of nutrition, students? They are chemoautotrophs. They can synthesize their own food with the help of chemicals. Chemoautotrophs. They are chemoorganotrophs with the help of organic material. Chemoorganotrophs. And they are also chemoautotrophs. Now, then the third one is respiration, student. Third one is respiration. They are obligate anaerobe. Now, what is the meaning of obligate anaerobe? Obligate anaerobe means they required anaerobic condition for their growth. In presence of oxygen, they will die. Obligate means strict anaerobe. Whereas halophiles are facultative anaerobe. Facultative, I have already told you the meaning of facultative means both aerobic and anaerobic condition they can survive. Okay. They are facultative anaerobe. Both are facultative anaerobe. Now, methanogens, student, with the help of carbon dioxide and formic acid, they are producing methane. And this methane is utilized for biogas formation. Biogas formation. So we can write some examples, students. One example is Methanobacterium. Okay, Methanobacterium. And the second example is Methanococcus. Example of methanogen. And in halophile, okay, suppose this is their cell wall membrane. And this is cell membrane. Okay, this is cell wall. This is cell membrane. Here some pigments are present. Here some pigments are present. The name of the pigment is Bacterial Rhodopsin. Bacterial Rhodopsin. Okay. Now they can synthesize ATP. This bacterial rhodopsin can synthesize ATP. This ATP will utilize for metabolic activity. Metabolic activity. They will not synthesize food with this ATP, or they will not help in carbon dioxide fixation. It will help in metabolic activity. And what about thermoacidophiles, student? Suppose in aerobic condition, okay, in aerobic condition, friends, in aerobic condition, sulfur is reduced to sulfuric acid. Okay. Now, in anaerobic condition, sulfur is reduced to hydrogen sulfide. I can give you some examples. Like one example is thermoplasma. One example is thermoplasma, and another example is thermoproteus. 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 So this is a comparative study of methanogen, halophile, and thermoacidophile. So with this, we have completed Monida, students. Next, I will start Protista. Till this, any doubt? Any one of you? Till this, any doubt? Any one of you? Students? Students, till this, any doubt? Any one of you? Students?
No, ma'am.
Any one of you? Any doubt? Okay. Now, Archaebacteria has some resemblance with eukaryotes. Okay. Now, what are the resemblance of Archaebacteria? Resemblance with eukaryote. There is a trick to remember, student. Okay, there is a trick to remember, that is M I MIL. What is MIL? Now, Archaebacteria used methionite. Archaebacteria used methionine. Okay, to produce protein. They use amino acid methionine for protein synthesis. Eukaryotes. Same in Archaebacteria also. Now, Archaebacteria contain some non-coding sequence. They are called introns. What are introns? Introns are non-coding sequence. Now, this non-coding sequence is also present in Archaebacteria. And L, their amino acid, only L amino acid is present. Now, in eukaryote also L amino acid is present. No D amino acid is present. D amino acid, L amino acid both are present in bacteria. Whereas in eukaryotes, only L amino acid is present. Okay, with this, we have completed our Monera part. So from today, I will start Protista. Okay. Protista. Now, what are Protista students? They are Protista are unicellular eukaryotes. Okay. Protista are unicellular eukaryotes. Now, what are the general characteristics of Protista? General characteristics. They are membrane-bound organelles are present. Membrane-bound organelles present. Okay. 80S ribosome. In bacteria, we have seen 70S ribosome student. 80S ribosome. That means resembles with eukaryotes. Because they are eukaryote. Now, what about locomotion students? Locomotion. 3 locomotory organ. One is pseudopodia. Second one is flagella. And third one is cilia. Now, their cilia and flagella contains 9+2 arrangement. Again, eukaryotic. And they are made up of tubulin protein. As in prokaryote, we have seen flagella is made up of flagellin protein. Okay. Then come to nutrition part, student. Nutrition. They are autotrophic. They are autotrophic. They are autotrophic. They are actually photoautotrophic. Okay, they are photoautotrophic. And second one is heterotrophic. Heterotrophic. In heterotrophic, you will get parasitic, symbiotic. Okay. And third one is holozoic or ingestive type. Or ingestive type. Then asexual reproduction, students. Asexual reproduction. Okay. Two mode of asexual reproduction. One is fission and another one is spore formation. Then sexual reproduction, mode of sexual reproduction. Sexual reproduction. First one is isogamous. Now, when condition is called isogamous, suppose this is one gamete, this is another gamete. Both gametes are morphologically similar. If two morphologically similar gametes are fused, morphologically similar gametes, they are called isogamous. The condition can be like this also, both are flagellated, like this also. Then condition is isogamous. Second one is anisogamous. Aniso means unequal. Suppose one is flagellated, one gamete, other gamete is non-flagellated. Okay. One gamete is of bigger size, one gamete is of smaller size. So we can say that morphologically dissimilar gametes. Okay. Dissimilar gametes. Then they are called anisogamous. And third condition is Eugamous condition. All these three types are present in Protista. And what is this Eugamous condition, students? Eugamous condition means, suppose I'm talking about male gamete and I'm talking about female gamete. Okay. Now, male gamete are smaller in size. Smaller in size. Okay. But female gametes are larger in size. Female gamete, reserve food is present. Reserve food is present. Here reserve food is absent. Okay. Now male gamete is large in number. Large in number. They are small in number. They are small in number. Now male gamete can be motile or non-motile. Okay. But female gamete is always non-motile. Now this protist is a link between students. This protist is a link between fungi, plantae, and Animalia. Okay. So let us see the division of Protista. Let us see the division of Protista. Now, Protista are classified into three categories. First one is photosynthetic. Second one is consumer decomposer category. Okay. And third one is protozoa. Protozoa, I will not teach you here, student. That I will teach you in zoology section. In zoology section, I will teach you this. Protozoa. So these two we will read here. In photosynthetic condition, number one is dinoflagellates. Okay. Now these dinoflagellate students, they show bioluminescence. Means bioluminescence means they emit light. Okay. One example I can give you. One example is noctileuca. They are also called night lamp. Now division student. Division of dinoflagellate is Pyrophyta. Okay. And class Dinophyceae. Class Dinophyceae. Second one is diatoms. Diatoms. Okay. Division Chrysophyta. They are also called desmids. Okay. Class Bacterial Features. Third one, Euglenoids. Division, Euglenoids. Class Euglenoids. Okay, this is the photosynthetic categories. Second category is consumer and decomposer. In consumer and decomposer category, student, example is slime mold. Again, slime molds are of two categories: cellular slime mold and acellular slime mold. Okay, now let us see example: slime molds. Why they are called consumer, student? Because they show holozoic type of nutrition. That's why consumer. Why they are called decomposer? Because they are showing saprophytic category of nutrition. That's why they are called decomposer. Now they are also called protistan fungi. Okay, protistan fungi. Now two categories this student. One is acellular slime mold and other is cellular slime mold. Two categories student. One is acellular slime mold and other is cellular slime mold. Acellular slime mold is also called plasmodial type and cellular slime mold is also called communal type. Some example of acellular slime mold: Physarum. Okay. Physarella. Euglenoids. Some example of cellular slime mold: Dictyostellum. Okay. Then Polyspondylium. Polyspondylium. Okay. Now we'll see some comparative study of Dinoflagellates, Diatoms, and Euglenoids. Okay. Firstly, we'll go with the photosynthetic category. For the first comparative study, students, we will see for Dinoflagellates. Okay. Diatoms and Euglenoids. Till this, any doubt, student? Any one of you? Students? Any one of you? Any doubt? Yes, Aditya, Agni, Sangamitra, Roshni, Riya, Shubhashri, Swaraj. Any one of you? Supriya, Tanmay. Any doubt, students? Any one of you? Till this? Roshan, Pujarani, Nilesh. Yes, students? Any one of you? Any doubt? Till this? Now let us see the comparative study for dinoflagellates. On the basis of habitat, they are mostly marine. They are marine as well as freshwater. and they are mostly freshwater. Come to its cell wall. Now they possess sculptured plate. Okay, their cell wall is made up of cellulose as well as pectin. Dinoflagellates are also called armored protists because they have structured plate. That's why sculptured plate they are called armored protists. Now their cell wall is made up of cellulose plus silica. Okay, their cell wall consists of two structures student like this. Soap box. Okay, they are called soap box like structure. This upper part is called epitheca and lower part is called hypotheca. Epi means above, hypo means below. Okay, and here cell wall is absent. They consist of pellicle. Okay, which are contractile in nature. Which are contractile in nature. So their pellicle is made up of proteins, lipids, and carbohydrates. What about their nutrition, students? We can say this is the first point of difference, habitat. Second, cell wall. Okay. Then the third one, student. Third one is students nutrition. Okay. Third one is nutrition. So they are photosynthetic students. They are photosynthetic. As in the earlier category, we have seen all are photosynthetic. Now, what are their pigments? Chlorophyll A, C, and alpha carotene and xanthophyll. These are the pigments. Here, chlorophyll A, C, beta carotene, and diatoxanthine. Okay. Now, in case of Euglena student, in presence of light, they are photosynthetic. Okay. Presence of light, they are photosynthetic. But when light is absent, they are saprophytic. Okay, so we can say that mixotrophic nutrition. We can say that mixotrophic nutrition in case of Euglena. What are the pigments? Chlorophyll A, B, beta carotene, and xanthophyll. Next, there is a food students. Fourth category. Their reserve food. Reserve food is carbohydrate. Carbohydrate plus oil. Now, in case of diatoms, it is vol. VOL. What is this vol, student? V for volutine granules. Now, volutine granules are responsible for storage of phosphate that I have already discussed with you in inclusion bodies. O for oil. Their oil content is so much high, so they float over water surface. So they float over water surface. Okay, and L for leukocyl. Okay, in case of euglenoids, they are made up of their storage material is paramylon. Paramylon. Fifth category is students, they are vacuole. Now, here non-contractile vacuoles are present in dinoflagellate. Non-contractile vacuoles. Okay. Their condition is that's why non-contractile This is condition called mesokaryotic because in eukaryote student contractile vacuole is present. Okay, mesokaryotic because large central vacuole, large central vacuole. Okay, eukaryotic condition students, and here also contractile vacuole, contractile vacuole. Okay, eukaryotic condition. Now here in dinoflagellates student, nucleus is large, large nucleus is present, large nucleus with histone protein students. Histone proteins are also present, but RNA is absent. RNA is absent. Till this, any doubt, students? So what we have discussed today, students? We have discussed the comparative study of Archaebacteria, that is Methanogens, halophiles, thermoacidophiles. Then the general characteristics of Protista, division of Protista, as well as comparative study of Dinoflagellates, Diatoms, and Euglenoids. In this, any doubt, students? Students, till this, any doubt? Students, any doubt? With this, any doubt, any one of you? Students? Okay, students, with this, I am ending today's lecture. Okay. Bye-bye, students.